Hello everyone. I have shared my screen. Can you all confirm that you can hear my voice and see my screen?
Yes, ma'am.
Okay. Okay. So this is the 10th TA session for drug delivery principles and engineering. And this week, the topics that were covered were based on vaccines, immune isolated cell therapy, and isolated cell and gene therapy. So based on these topics, we have some previous year assignments and some questions that I got. So we'll go through those questions. Here is the first question. In immune isolated therapy, the cells should be within dash from the nearest blood vessel to facilitate effective diffusion of nutrients. What should be the distance of these cells from the nearest blood vessels?
500 micrometer.
Okay.
Option A.
Okay. Anybody else would like to answer? Yes, both of you are correct. The correct answer is 500 micron. So if the cells are within 500 micron from the nearest blood vessels, it will help in effective diffusion of the nutrients. Okay, we have our next question here. Which of the following would antagonize immune isolated therapy? You have to select two correct options here.
Second option and the last one.
Okay.
Option 2 and 7.
Okay. Anybody else would like to answer? Anyone else? Which of the following would antagonize immune isolated cell therapy? Okay, the correct answer here would be option 2, use of biodegradable polymer to encapsulate cells. And second last option, use of polymer that allows free exchange of proteins and cells to encapsulate therapeutic cells. So immune isolated cell therapy is basically you are capturing these cells in a non-degradable polymeric cage like thing so that the cells are isolated from your system, but then they will release nutrients. They are free to get nutrients and water and oxygen from your body, and they'll release therapeutic proteins which is eventually help you in the therapy. But here, if you see the last option that someone has chose, use of polymer that allows free passage of small proteins, um, but not cells and larger complexes. This is true. So basically, you're asking antagonize. So we have to select something opposite to what immune isolated cell therapy is doing. If you use biodegradable polymer to encapsulate these cells, then it will degrade the cage basically, and your cells are free to move out, which basically is opposite to what we want in the immune isolated therapy. And also use of polymer that allows free exchange of cells, proteins and cells. So along with proteins, your cells are also since free to exchange. This is also defeating the purpose of immune isolated therapy. So both of these options will be the correct answer. Okay, moving on to the next question. The process of weakening a pathogen while generating a vaccine is called.
Option 2,
Attenuation.
attenuation.
Okay,
Option 2.
okay,
Attenuation.
okay. Okay, so this is a straightforward question, and all of you are correct. It is attenuation. So, attenuation is the process of weakening the pathogen to generate a vaccine. Okay, we are on to our fourth question now. What are the major challenges of immune isolated cell therapy? You have to select two correct options here. Major challenges.
Option 1 and 2.
Option 1 and 2. Okay, anyone else? What are the major challenges associated with immune isolated cell therapy?
Option 1 and 2.
Okay, Anybody else would like to answer? Yes. What are the major challenges of immune isolated cell therapy? Anybody else would like to answer? We have to select two correct options here. Okay. So option 1 and 2 would be the correct answer. Generation of immune reaction and fibrosis of implant over an extended period. So immune isolated cell therapy is basically preventing an immune reaction, thereby providing therapy. So the generation of immune reaction is one of the major challenge. And fibrosis of the implant is basically when your implant gets covered with immune cells as well as their secretions, and it covers the entire implant. So if it covers it, the released products from the cells will not be able to diffuse out. So that poses another challenge in immune isolated cell therapy. So both option 1 and 2 would be correct. Okay, we have our next question here. Which of the following methods can be used for encapsulating cells in biomaterial? You have to select two correct options here.
Ionic gelation and microfluidics.
Okay, what about others? Which of these methods you think can be used for encapsulating cells? Anybody else would like to answer? Yes, ionic gelation and microfluidics would be the correct answer. So in ionic gelation, you are basically using two different solutions, one polymer solution and one ionic solution to create an interaction between these that will form a gel. And in microfluidics, you can, similar to this, you have your polymer solution and your cells and some cross linkers. So if you flow them through the microfluidic channels, at the end you'll be able to encapsulate that. But in others, spray drying, hot melt, all of these, heat is involved that might be detrimental for cells. And in emulsion based methods, it uses high mechanical agitation to create emulsion that can be also disruptive for cells. So other methods cannot be used, and ionic gelation and microfluidics are the appropriate methods for encapsulating cells. Okay, we have our next question here. What type of vaccine is given before exposure to a disease?
Option C.
Okay,
Option 3.
option 3. Okay, number 3. Okay, anybody else would like to answer? What type of vaccine is given before exposure to a disease?
Option 1.
Option 1. Okay,
Sorry, option 3.
okay. Anyone else would like to answer? Okay, the correct answer is option 3, prophylactic vaccine. So, prophylactic is the vaccine that is given before exposure to a disease, and this post exposure prophylaxis is between exposure and actual development of the disease in that time period, and therapeutic vaccine is given after the disease has developed. So, option C, post prophylactic vaccine is the correct answer. Okay, we have our next question here. Which branch of immunity is triggered when a vaccine is administered to an individual?
Option 1 and 2.
1 and 2. Okay.
Humoral and adaptive.
Okay. Others?
Option 1 and option 2.
Okay. Yes, all of you are correct. Humoral immunity and adaptive immunity are triggered when a vaccine is administered to an individual. Moving on to the next question. What is an adjuvant in the context of immune response?
Option 2.
Okay.
Second option.
Okay. Others? What is an adjuvant in the context of an immune response?
Option 2.
Okay. Anyone else would like to answer? Second one. Okay. Yes, all of you are correct. A molecule that stimulates the immune response to target antigen is basically an adjuvant. Okay, we have our next question here. Why is it important to ensure that the thickness of the membranes in immunoisolated implants remains within a certain range? For example, 200 to 500 micron.
Option B.
Okay,
Option 2.
okay. Anyone else?
Option B.
Okay. Yes, all of you are correct. So we have this certain range of thickness to ensure proper diffusion of oxygen and nutrients across, and it prevents immune cell entry into the encapsulated polymer cell encap in inside the implant where cells are encapsulated. Okay. We have our next question here. Which of the following are undesirable properties of a polymer used for immuno-isolated implant?
Option 4 and 5.
Okay.
Option 4 and 5.
Okay. Others? Three and four. Okay. Anyone else would like
First and
to?
fourth.
First and four.
Yes, ma'am.
Okay. Okay. So the correct answer would be option last two options, toxic and biodegradable. So here we are talking about undesirable properties of a polymer used for immunoisolated implant. So if it is toxic, then obviously it will be detrimental to the system. If it is biodegradable, it will release the cells that we have encapsulated into our implants. So that defeats the purpose of the immune isolated implant, where we specifically capture the cells, encapsulate them into an implant so that they are isolated from the body, but then they can release therapeutics. So, if you are making it non erodible, that is a desirable property. So, it should be non erodible, biomechanically stable, and biocompatible. The undesirable properties would be toxic and biodegradable. Okay, we have our next question here. Which of the following is a potential disadvantage of particle based approaches while delivering vaccine to antigen presenting cells? You have to select two correct answers here.
Last two options.
Okay. What about others?
Last two options.
Okay. Anybody else? Which of the following is potential disadvantage of particle based approaches while delivering vaccine to APCs? Anybody?
Four and last.
Four and last. Okay. Okay, so here the correct answer would be last two options. The formulation can be unstable and the antigen might lose integrity in the process of encapsulation. These are the potential disadvantages. The formulation can increase the in vivo half-life of the vaccine, is basically an potential advantage of particle based approach because it will reduce the number of times you are giving the dose of vaccine. Okay, so these two will be the correct answer. Formulation can be unstable and antigen might lose integrity in the process of encapsulation. Okay, we have our next question here. Why is polyethylene considered toxic in gene delivery applications and what measures can be taken? Uh, Ignore the what measures can be taken, just the reason why it is considered toxic in gene delivery.
Option B.
Okay, what about others?
Last option.
Okay, anybody else?
Option 2.
Okay, okay, the correct answer would be option 2. Its cationic nature leads to cellular toxicity at higher doses. So polyethylene can be used for gene delivery. It can easily pass through the cell membrane, but at higher doses, because it is cationic, it will interact with the negatively charged components of the cell, and that is how it leads to cellular toxicity. And talking about what measures can be taken, you can basically uh, mm, use reduce the charge by using something else, or reduce the length of the polymer chains to reduce the number of cationic ions that is interacting. And you can conjugate other molecules or basically modify it to reduce this cationic nature so that it's less toxic at high doses. Okay. We have another question. What is the main advantage of using non-viral vectors in gene therapy over viral vectors? What is the main advantage of using non-viral vectors in gene therapy over viral vectors? Yes, anybody?
Option C.
Option C. Okay. What about others? What is the main advantage of using non-viral vectors? Okay. Option C is the correct answer, low immunogenicity and easier mass production. As compared to viral vectors, this is the only main advantage. Otherwise, viral vectors have natural tropism and higher transfection efficiency, and they integrate into the host genome easier than the non-viral vectors. Okay, these were all the questions I had for today. If anybody has wants me to go through any question again, or people who joined later, if you want me to go through any questions again, let me know.
Hello, ma'am.
Yes,
Excuse me, ma'am.
yes.
Ma'am, on which date that exam will be conducted?
On which date this final exam for drug delivery, you mean?
Yes, ma'am.
Actually, I have not been notified by that exam. Oh no, wait. I think 23rd October, 23rd or 20. Yeah, around that time, if I'm not wrong. But you'll receive a mail regarding this.
Okay, ma'am. Ma'am, little problem is there. May I change the center now?
That you have to mail the NPTEL people. They handle all these things. You have to mail them and ask if you can change.
Okay, ma'am. Thank you.
No problem. Anybody else has any other questions or doubts or wants me to go through any question again?
Thank you, ma'am.
You're welcome.
Thank you.
You're welcome. You're welcome.